Welcome, everyone, and thank you for joining us for our webinar titled Functional Ultrasound Imaging in the Brain of Awake Behaving Mice. This is Andy Henton from Inside Scientific, and I'm very pleased to be your host for today's event. Our session is sponsored by Neurotar and Iconius, and will feature Dr. Zsolt Lenki, Research Director at INSERM in Paris, and Dr. Artem Shatilo, Head of MRI at Charles River Laboratories in Finland. Today, Zsolt and Artem will discuss how functional ultrasound imaging in awake and head-fixed mice can help advance basic neuroscience and neurovascular research, as well as preclinical drug discovery. And with that, I'm pleased to welcome our first presenter, Dr. Zsolt Lenki. Zsolt, thanks for joining us today. Please feel free to begin your presentation when you're ready.
So, thank you. Thank you for the opportunity to discuss about our research, and thank you for everybody listening to us. So first I have to make a disclaimer. I'm co-founder, stakeholder, and advisor at Iconius. So having said that, this is the outline of my talk. So first I will present a bit the technology behind FUS imaging and how we adapt it to awake mice brains. Next, I will present a recent publication of our team about functional imaging of the default mode network in mice. And finally, I will show you some unpublished data about how to use FUS imaging to image pharmacological action on brain activation and connectivity. So first, let's look at the main preclinical functional brain imaging techniques. The gold standards are fMRI and PET. Optical imaging has some advantages for imaging. So basically all these techniques have their own advantages and disadvantages, but any of them is really perfect. So there is another way to looking at the brain, and this is by using classical Doppler ultrasound. But this classical way of doing ultrasound imaging has limited sensitivity. So in the last decades, there was a research team in Paris at the ESPCI Paris Engineering School that proposed a new way of doing ultrasound imaging, but they started to call ultrafast ultrasound. And this has a huge promise of overcoming the poor sensitivity of Doppler ultrasound. So how does it work? So as it was shown by Michael Tanter and collaborators, if you compare this technique to conventional imaging that you see on the left, where you scan the tissue with focused acoustic waves, in ultrafast imaging, you use multiplane waves that resonify the tissue at the same time, get back much more information from the tissue, and finally it will lead to a much better signal-to-noise ratio, as you can see if you compare the quality of these two images. So being much faster, typical imaging times are orders of magnitude better or faster by using ultrafast imaging than normal classical ultrasound. So this leads to up to 100-fold increase in sensitivity, which allows us to image slowly moving blood in small arterioles, where most of the neurovascular coupling takes place. It has an excellent spatial resolution, especially as compared to fMRI. It's 100 micron or less in plane, and it's very sensitive to detect hemodynamic changes of a couple of percent even without averaging. So that allows us to image brain activity through the neurovascular coupling. So here you see an example for that. So whisker stimulation through the intact skull and skin in mice, and you see that stimulation of whiskers leads to elevated blood flow in the contralateral parietal cortex. So another way of looking at brain activation is mapping brain connectivity. So this is a technique that was invented first in fMRI, where researchers observed that you can map correlations in low frequency spontaneous variations of blood flow. And by using these techniques, you can add to the classical anatomical networks that you see on the left, you can add functional connectivity networks. And in fMRI, people call these networks resting state brain networks as well, because basically they just ask the subject to lay quietly in the fMRI machine. And mapping correlations draws us the classical sensory or motor networks in the brain and some other networks that you really see well in the resting head, which is, for example, the deaf hormone. So we are using this technology now by using this specific small ultrasound probes in mice, which have typically 15 megahertz central frequency that allows us to see the mice brain in whole depth with optimized resolution. And we fix this probe in the head of the mice. Sorry, I'm coming back to the video. By applying, by surgery, chronically a small metal frame, and we can clip on the probe on this metal frame. So here you see the mice with the metal frame on the upper left panel. On the upper right panel, you see how we can fix the mice into the mobile home cage that we use typically for health fix configurations. And the bottom left panel, you see our typical habituation protocol, where 2 days after surgery, we start to handle mice. We do 10-minute habituation, followed with some handling. Then we follow up with handling, a bit more habituation, and finally, at the third habituation day, we do half an hour of habituation. And this allows us to have relatively quiet and unstressed mice. So here you see the mice walking in the mobile home cage. And in the awake mice brain, then we map correlations of spontaneous variation of blood flow. So here you see 2 examples of region of interest pairs. So on the upper panel, you see the contralateral homologous parts, homotypic parts of the barrel cortex in blue and red. And you see this is quite highly correlated, while the barrel cortex is not very well correlated with the retrospinal area that you see on the lower panel in green. So by using these regional comparisons, you can establish connectivity matrices where highly connected regions are labeled in red, while lowly connected regions are labeled in blue or green, as you see in this example. So by using this technique, we first checked literature data that suggested us that functional connectivity patterns are highly sensitive to anesthesia. So you see the connectivity matrices here from a group of mice without any anesthesia first on the left. Then by using a very slight dose of sedation, which doesn't change as much connectivity, then you see a typical anesthesia dose, and you see that correlations are significantly weakened. And this weakening is really related to the consciousness or the awakeness level of the animal, because if we inject antipamazole, antisedant to wake up the mice, immediately you get back the full strength of connectivity. So it confirms that mapping connectivity in the mice, at least, is really very sensitive to anesthesia. So by using then this technique, either in fully awake configuration or the very light sedation, we can look at the connectivity patterns. So This is another way to look at connectivity. This technique is, or similar techniques are often used in fMRI as well. For example, there are several sorts of independent component analysis that allows you to obtain this data. So here we used unsupervised spatial singular vectors that show overlapping networks at the same coronal plane. So the first spatial singular vector, you see the strong bilateral connectivity of the cortex through the corpus callosum. On the second spatial singular vector, you see the strong connectivity of this midline default mode network related structure, which is anticorrelated with more lateral somatomotor regions labeled in blue here. On the third vector, you see the connection of the default mode network with the striatum. And on the first one, you see the connection of motor cortex with the striatum. And actually, these networks correspond to real anatomical networks, as it can be seen on the right, which is a classical anatomical tract tracing experiment, where a tract tracer virus was injected to the neighboring anterior cingulate cortex, default mode network component, or to the bit more lateral primary motor cortex, and you see that the tract tracing highlights the same contrasted connectivity patterns that we see in live by using FUS imaging. So in the mice, now we have some new unpublished data on that. So here you see average patterns from 50 mice, where you see also very highly contrasted networks that respect well anatomical boundaries. So it's a proof that the connectivity that we measure is anatomically relevant. So we typically overlay the average vascular image with the connectivity image and the atlas image, and finally it allows us to really see in detail how the connectivity pattern corresponds to neuroanatomy. So this is a magnified example for that. So to summarize, FUS provides anatomically and functionally relevant activation and connectivity patterns, as it was shown by respecting anatomical boundaries and the response of the barrel cortex to whisker stimulation, for example. It has a very high spatial temporal resolution. Imaging is very high speed that allows effective filtering of physiological noise. All this together leads to a better imaging quality in resolution and signal to noise ratio than fMRI. Very importantly, we don't absolutely need anesthesia or immobilization, so it can work in awake and behaving animals. And last but not least, it's much simpler and cheaper to use than fMRI. And finally, as we published a couple of years ago, the same technology allows super resolved imaging of the vasculature that could interest people that do research on brain vasculature. So now I will get to the second part of my talk about the functional imaging of the default mode network in mice. So first, I present the default mode network. So this is a very special network that is stronger and more activated and more strongly connected in subjects in the fMRI machine that are just completely relaxing, not doing any specific tasks. And when the subject engages in a sensory or motor task, the default mode network is deactivated. But actually, this deactivation of the default mode network is impaired in different pathologies like autism, schizophrenia, or Alzheimer's disease. So it would be very interesting to reproduce this default mode network and this deactivation in preclinical models, and mostly in mice, which is the major one of the major preclinical models. But until now, maybe due to limited sensitivity of of existing techniques, it wasn't proven that in mice you can find similar task-induced deactivation of the left hormone network. So this is what we try to check. There is anatomical proof for deaf hormone network, for the presence of deaf hormone network in different models, like here you see in rats, monkeys, and humans, and also in mice, but this is just anatomical in mice. So we looked at the brain connectivity, first brain activation here, of mice in the mobile home cage. Here we used a very light sedation that just slowed down a bit the spontaneous movement of the mice in order not to have too many motor activity-induced activation of the brain, but the mice were awake and responding. So you see that there is expected, on the very left of the graph, there is an expected activation of the contralateral barrel cortex, and interestingly, we find significant deactivation of the deaf hormone network component, retrospinal cortex. So this is a first proof that there could be task-related deactivation of this major hub of the deaf hormone network. And then we looked at connectivity, and similarly, while we have expected loss of connectivity between the two contralateral barrel cortexes during activation, because the activation is unilateral, we have also a significant disconnection of the two deaf hormone network hubs in this coronal section. So this data we consider as a proof for the functional existence of deaf hormone network in the mouse brain. So here, for the first summary, we validated the mobile home cage for the imaging of sensory processing in awake mice. Whisker stimulation leads to deconnection of the barrel cortex, and also to elevation of bilateral connectivity hippocampus, another major hub of tactile information processing. Importantly, we find significant deactivation and disconnection of the mouse retrospinal cortex, which corresponds to the posterior cingulate cortex in humans, which is a major hub of the deaf hormone network. So let's get now to the second part, which we tentatively call pharmaco-fus. We did here a proof of concept study with the aim to quantify pharmacologically-induced dynamic changes in brain activation and connectivity in awake mice by FUS. So for that proof of concept study, we used a classical pharmacological agent, scopolamine, which is a muscarinic receptor antagonist, and it's a widely used model to model cholinergic system alteration, which you could find in clinics in neurodegenerative disorders like Alzheimer's disease or Parkinson's disease. So we asked this simple question: does scopolamine injection lead to time and dose-dependent hippocampal disconnectivity? Because that would be a first proof that FUS is capable to produce pharmacologically relevant connectivity patterns. So here you see the experimental setup. So we did two short periods of baseline imaging, 2 times 10 minutes. Then we injected first 3 milligram per kilo scopolamine, and we continued with these 10-minute registering periods. And on the upper row, you see a seed placed in the left hippocampus, and you see that after scopolamine injection, there is a much higher bilateral connectivity in the hippocampus, and you see a similar result when the seed is placed to the left somatosensory cortex. After scopolamine injection, there is higher bilateral connectivity in the cortex. So now, if you compare several regions of interest on the bottom, you see that there is a general elevated connectivity after a scopolamine injection. So we did this first series of experiments in a relatively small group of mice, and analyzing this connectivity matrix with a classical statistical test like a bone pharaonic correction really did not allow on this very small first group to have, we have seen clear tendencies statistically, but it did not give in this small group statistically relevant changes. And we were, sorry, I get, yeah, sorry. And we were thinking that as we have a very readily recognizable pattern on this connectivity, whether by training a machine learning algorithm would allow us to classify treated and non-treated animals by showing that you can use really small group sizes to see pharmacological relevant changes in connectivity. So we trained the machine learning algorithm to construct this ad hoc pharmacological score that we call the scopolamine score in this study. And we did it from connectivity patterns of 5 scopolamine treated versus control mice. And then we tested the performance of this algorithm in an independent cohort of 8 mice, where we got a very strong classification or discrimination of treated and non-treated animals. So showing that the pharmacological score is sensitive to detect the pharmacological action. This is a classical validation of the method by cross-validation that led to this refined weight metrics. And it shows that this method has very high accuracy, sensitivity, and specificity. So this next question was whether this pharmacological score is pharmacologically relevant. So we looked at how the scores change in dependence of the scopolamine dose, and we have found dose-dependent increase of cortical and hippocampal connectivity. Another pharmacological control was reversal by milamelin, which is a muscarinic receptor antagonist. So this leads to a pharmacologically antagonist action of scopolamine. And finally, we have investigated whether it was a real brain-dependent effect or not a peripherically induced effect. And we used a pharmacological analog of scopolamine, methylscopolamine, that doesn't crosses the blood-brain barrier. And as you can see on the very right group of graphs, methylscopolamine did not lead to the pharmacological score. So for that part, the summary proposes that our discrimination model shows important scopolamine-induced increase of hippocampal cortical connectivity. This pharmacological score led to a robust discrimination of scopolamine treatment from baseline in independent dataset. And in another independent group, the pharmacological score was found to be dose-dependent and dependent from penetration of scopolamine into the brain. And I don't show this diapositive here, but one of the advantages of FUS is at the same time that you measure neurovascular coupling, you measure blood volume. And we have also shown that this change on connectivity is not related to global changes of blood flow. So if I get to my final conclusions, FUS imaging provides very highly resolved, anatomically and functionally relevant activation and connectivity patterns in the awake mouse brain. We validate the mobile home cage for FUS imaging of sensory processing. in awake mice. By using this tool, we provide a first functional evidence for the default mode network in the awake mouse brain. And finally, we introduce pharmaco-fus as a quite simple, robust, specific, and sensitive tool to monitor drug effects on perfusion and functional connectivity at the same time in the awake mouse brain. So this work was done in close collaboration with Professor Michael Tamper from the Physics for Medicine Paris lab. And in my team, Andrea Cleaver, Jean-Charles Mariani, and Jeremy Ferrier, who was a postdoc in my team, and now he's application specialist economist. These were the three persons that contributed importantly to the data that I have shown you today. Thank you for your attention.
Thank you, Zsolt, for this excellent presentation. And up next now, I'd like to introduce our second presenter, Dr. Artem Shatilo, who will discuss how Charles River Laboratories in Finland uses functional ultrasound alongside MRI to support Charles River's functional neuroimaging platform. Artem, it's great to have you with us, and feel free to take it away whenever you're ready.
Thank you, Andy. Thanks for the introduction and having me, and also thanks, Zsolt, for the great presentation preceding my talk. So, as you can hopefully see, Charles River is not only the mice and rats company. So our Charles River site in Kuopio is part of Charles River Laboratories Discovery Branch, which is a full-service preclinical CRO for drug discovery and development. And here in Kuopio in Finland, we are focused on CNS and neurology, running studies in disease models in all the therapeutic areas you can now see on your screen. In practice, what we do is we bring or generate a model of certain disorder, and then we combine a panel of behavioral tests, biomarker assays, and of course imaging in the same animals under the same roof of our facility. And when it comes to imaging, we are running a full suite of 2 high-field preclinical MRIs, nuclear imaging lab, and now, thanks to our collaboration with Iconius, we have functional ultrasound prototype system with Neurotar's mobile home cage, which I'm now going to tell you about. As you know, those of you who work with imaging, conventional imaging modalities, they can deliver a lot of useful information, from anatomical integrity to molecular composition of the brain structures, and also, very importantly, how the brain structures function and how they communicate together. As Zsolt mentioned, golden standard for this has been fMRI, also because of its translational value to the clinics, and it offers this wonderful whole brain functional coverage, great contrast, target engagement, PD data, a lot of visual and statistical information that can support your research question. But at the same time, especially in animals, it is a highly complex procedure that demands a lot of skill and knowledge, and it starts from surgery, all the features of anesthesia, physiology maintenance, proper acquisition, and then it doesn't stop there. It goes to the data handling and proper analysis. And at the moment, it is mostly limited to acute imaging in rats, and it is highly challenging in mice. And when it comes to awake imaging, there has been some research, but it's still a large R&D area, and it's not very practical to perform in industrial setting. So in my opinion, functional ultrasound is a new technique that could actually address most, if not all, of this limitation. In functional ultrasound, even though the readout is different, similarly to functional MRI, the whole panel of stimulation paradigms can be employed to test the sensory pathways, such as mechanical whiskers, or let's say hind limb stimulation, or visual stimulation with light of various frequency and intensity. And the examples of the data you can see on your displays right now. And for example, whisker stimulation has been used in our lab to test for abnormal sensory processing in the trigeminal system in OIPN, which is oxaliplatin-induced polyneuropathy model. That model is based on well-known side effect of chemotherapy drug oxaliplatin, which causes, among other things, numbness in the face and loss of lips sensation in oncological patients that receive it. So when we measured neurovascular response to whisker stimulation in these mice with functional ultrasound, we found that RCBV change is actually smaller, significantly smaller in animals with polyneuropathy compared to controls, which is well in line with the clinical manifestations. And besides physical stimulation, we are, of course, very much interested in detecting changes induced by pharmacological agents. Like in this case, we used functional ultrasound with intravenous injection of small dose of nicotine, kind of reference compound, to test the responsiveness in Q175 Huntington disease model, and found quite major differences in the response profile. And this figure also highlights nicely the importance of high temporal resolution, because it is possible to see and examine each stage of the response from this sort of initial dip, the decay of the signal, and peaking of the response. Then in our Button's disease model characterization work, in CLN8 mice, we used functional ultrasound with visual stimulation of different intensities to address this major impairment of the visual system and actual retinal atrophy in these animals. And at 8 months of age, we found that transgenic mice had absolutely no response to visual stimulation of low intensity, while with higher intensity, up to 60 lux, it was still possible to get some activation in the visual system in superior collicles, for example, and that was a very prominent effect that kind of contributes to the model characterization and the panel for the future drug testing. Then, even without the stimulation, it is possible to use functional ultrasound to look at the differences in resting state functional connectivity, which can be particularly useful in neurodegenerative disease models, such as, in this case, it's P301S, tau Alzheimer's disease mouse model. And we found much lower connectivity values pretty much across the brain. And then even kind of going against the method's name and using functional ultrasound in a structural mode only, it still provides quite a bit of value and interesting information, for example, in stroke or oncological models, being able to show this kind of vascular remodeling processes and perfusion deficits, which is when combined with non-invasiveness and possibility to do repeatedly, the progress can be tracked and observed throughout the development of the pathological process. And overall, functional ultrasound having few limitations, such as inability to get whole brain data as we are used to in magnetic resonance imaging, or difficulties with this kind of cross-modality co-registration, as it's not showing the usable structure for that registration. It still provides quite unique advantages, such as very high cost effectiveness for awake imaging, which is particularly important in our setting when we have to massively work with animals in high throughput mode. And you can combine that with the locomotor tracking, thanks to the feature in NeuroTAR's mobile home cage. And now on your screen, you can see at the same time how the animal is behaving, the stable signal on the screen that we're getting from the functional ultrasound, and the tracking that we are recording simultaneously, synchronized with our measurements. And then, of course, the very practical things, such as small system footprint and possibility to integrate other readouts, such as, let's say, electrophysiology or implanted optogenetic probe, or something else that you are interested in, it gives you this huge and broad spectrum of applications that is we are hoping to apply in our work across all the Charles River library of animal models. And I guess this is all I can cover now without taking too much of your time, but I hope to tell you more while answering the questions. Thank you. Andy, back to you.
Thank you, Artem, for your presentation as well. And with that, let's move on to our Q&A session. Okay, so let's jump right in with the first one. And actually, I'm going to ask you to cover this, Zsolt. What is measured exactly by functional ultrasound?
Okay, so in this implementation that I was showing, functional ultrasound measures directly the quantity of red blood cells in any given voxel. So functional ultrasounds measures directly cerebral blood volume.
Okay. Excellent. And maybe as an extension of this, why is it important to have such high temporal resolution?
So actually, this high temporal resolution comes naturally with the technique. This is not a point that was particularly researched to push even further, but as we had this very high temporal resolution, it allows us to have really a lot of data, and this data allows to do some advanced filtering mechanisms, processing. So finally, in the end, it comes down to a much higher signal-to-noise ratio that we could have had without having such high temporal resolution. So this is a clear advantage of the method. And of course, this high temporal resolution allows us to filter out directly lower frequency physiological signals like cardiac or respiratory related data that can be a nuisance factor, for example, in fMRI imaging, much more complicated to filter out. Sorry for that. So I'm finished.
No, that's an excellent answer. Thank you. I'm sure that covers it. Next question here. How long does it take to make a reliable measurement? And I think in this context, we want you to comment on both the setup of the mobile home cage as well as the functional ultrasound procedure itself. Artem, could you perhaps take the lead on this one first?
Sure. So there is no straight answer because it depends very much on the type of the study, type of the measurement, and the overall plan. But in practice, once the whole system has been set up and there is a standard operating procedure and you have trained people to do that, it takes a maximum of 10 minutes from the anesthesia or whatever the preparation of the animal you're doing to actually getting the scanner going and acquiring the data. And then, of course, depending on what you want to measure, if it's the pharmacological session, it's typically a little bit longer. But if it's the one of those sensory stimulation scans, which I showed, those are typically very short, from 5 to 7 minutes max.
Excellent, excellent. And Zsolt, in the applications, you're running in your lab or through your experience, in your opinion or in your experience, how long does it take to make a reliable measurement?
Yeah, so in order to have a reliable functional connectivity data, you need a couple of minutes, up to 10 minutes continuous recording. So as you have seen in the scopolamine paper, we used several successive 10 minutes periods. And this experiment, for example, would last for typically around 1 hour. So for us, a typical pharmacological experiment is in the 1 hour range. Sensory stimulation, as Artem said, can be much quicker. So, or typical whisker stimulation protocol during our typical whisker stimulation protocol, we are stimulating the whiskers for 30 seconds, then we wait 60 seconds, then we do it again, and we do it 10 times, and from each stimulation period, we typically get significantly higher blood flow in the barrel cortex. So then it depends how much data you need, but as Artem told, typical sensory stimulation protocols can be quite fast. You can start imaging just after minutes after setting up the system, and you see the data almost real time on the screen. So this is something which is very straightforward and rapid.
Fantastic. Those are great insights. And actually, let's stay on this theme of making reliable measurements and going through the methodology and getting things prepped. Can you both comment on habituation? So, Zsolt, you had a brief moment in your presentation where you touched on this, but maybe could you elaborate on the habituation procedure for the animals when you're using the mobile home cage and the Iconius imaging device?
So we found that habituation is relatively short. So I have shown that we have 3 following days when we do each time 10 to 30 minutes habituation. So the handling time for this mice is quite short, and we think that it allows us to image them nicely habituated and relatively stress-free mice. So compared to other head-fixed systems, I consider it as a quite short habituation protocol. Of course, it needs some regularity. It's better to handle the mice the same time of the day, and then you will do the experiment. Of course, it's better if the same person handles and habituates the animals that use the experiment. So, but these are just classical behavior methods.
Yes, no, those are great insights, and yeah, that's fantastic. Artem, what about you? Just anything to add there about habituation and?
Sure. Well, we are using a little bit more extensive habituation paradigm. So in our license and procedures, we are allowing our animals to recover from the head plate implantation surgery for about a week. And then we are trying to time the whole process into 1 working week, so like 5 days. And we start on Monday with very short just handling and habituation to the operator. We're using the same technician, same person, training the animal and collecting the data in the end. And then we're day by day increasing the handling and the training time in the mobile home cage with the system all the way till Thursday, up to the maximum duration of 45 minutes per experiment. And then on 5th day, on Friday, we are actually scanning them. And that gives pretty nice results.
Oh, excellent. Okay, thanks for sharing that specific protocol. Let's shift gears here. Here's a great question about ultimately deciding on the appropriate device or method to support FUS imaging. So for each of you, and maybe I'll start with you, Artem, why was it that the mobile home cage was maybe the appropriate device to use instead of an alternative solution, such as an airlifted ball, a treadmill, or a disc? I mean, in your case, what made this the right partner for FUS imaging?
Right. So we found that those major advantages that mobile home cage offers are those, first, the compact size, the ability to do training in a separate cage, having 2 of them, similarly training animals in one and then measuring them in another. It allows you this parallel kind of processes, which accelerates the whole study, and then of course the freedom of movement and the openness and the possibility to combine with other devices such as stimulation or tracking feature. Those make it pretty much the main choice for us.
Excellent. And Zsolt, what about you? And what was your discovery process of utilizing the mobile home cage for your research?
Yeah, actually, I would just add, I completely agree with Artem on that. I would just add maybe 2 additional points to that. So, as compared to the airlifted ball method, so I really think that mice are feeling themselves much more comfortable in a dark box where they can touch the side of the box, the wall. They can have immediate tactile information feedback, and they are just more comfortable, I think, in a box than running on top of a ball, where they have a visual in a typical floating ball configuration. They have a visual information around, but mice are not so visual animals. They are much more tactile animals. So this is one point. And the second point, that at least as compared to the typical commercial floating ball configurations, the mobile home cage, at least at the time when we started to work with them, it was much cheaper. So it's a simpler system. It seemed to be more robust for me. So these were the additional components that I would add to what Artem told us.
Great responses. That's helpful. Very good. Okay, another question here, and Artem, I'm going to ask you to take this one because you brought up stimulation protocols actually just when we were addressing that last question. So what types of stimulation can be used with functional ultrasound imaging?
Yep. So as I mentioned, we started with the sort of standard set that has been used also in our labs in functional MRI setting. So it's the sensory stimulation of any available body part of the animal. Thanks to the kind of accessibility and openness of the setup, you can stimulate whiskers, ears, paws, whatever you name it. And then, of course, the pharmacological stimulation is the number one interest for us. And then, of course, it just comes down to your creativity and ability to manufacture those additional stimulation devices, such as guided lights for the visual stimulation, or let's say thermal stimulation with lasers. It's almost not limited by the features of the system itself.
Excellent. Thank you. Great answer. Another one here. Is FUS limited to 2D, or can you image in 3D as well? Maybe this would be a good question for you, Zsolt.
Yeah. So in most current papers of FUS, it's a 2D method by using a linear array and imaging at any single time one slice of the brain. Recent technical development opened other dimensions now for FUS. So you can have basically 2 technical approaches to do 3D FUS imaging. So either you move this linear probe by a motorized setup, for example, or you turn to make tomography like images, or you are using a matrix array, a 2-dimensional matrix array that acquires at any given moment 3 full 3D volumes of the brain. And this was published last year by Michael Tantas in a very nice Nature Methods paper. So both methods have advantages and disadvantages. I wouldn't go really into the details that we detail a bit in a recent Iconize newsletter. I would say that currently the matrix approach is much less sensitive than the linear array. So currently our preferred setup is the motorized 2D probe. And but in the future, probe technology will develop, and snapshot 3D or 4D volume imaging will be clearly the future. Just in the very in the next years, maybe it's better to use motorized linear arrays to obtain 3D data.
Fantastic. This is a great answer. Thank you very much. Okay, I think we've got time for maybe one or two more questions. So let's, hopefully this is a straightforward one to answer. Can these measurements, these procedures that have been shared be done in awake rats? Do either of you have experience working in awake rats? Artem, maybe
Zsolt?
I'll ask you that.
Maybe I will jump in. Sorry. Sorry, Artem. I will start with that first, maybe. So our lab did not do awake animals, but our colleagues in Paris did it. So actually, you can use it to detect brain activation and perfusion in freely moving rats, as it was published by Ivan Cohen's lab very nicely. So it's fully possible, of course, to adapt FUS technology basically to any awake animal type. So mice are quite small. So and the signal to noise ratio in the brain, in the very small brain, is quite low. So I would say that if you can do it in mice, then you will be able to do in most typical preclinical models that are bigger than mice.
That makes sense. Thanks for confirming that. And actually, let's squeeze in one more question. And it is pharmacological stimulation or compound dosing possible in the mobile home cage in awake animals? And I think the answer is yes, but Artem, maybe you can just take the lead on this one and elaborate about your experience.
Yes, the answer is definitely yes, but it is, of course, a little bit special procedure. Of course, while animal is awake and behaving, you don't want to disturb or stress it out with any sort of injections and this kind of rough handling that is sometimes required for the dosing. So what we have been exploring is the use of vascular access ports, so pre-implanted device that lets you inject the compound into intraperitoneal or intravenously or subcutaneously through the sort of port that is protruding from the skin. So the injection itself does not disturb the animal at all. And when you are setting up the functional ultrasound management imaging session, it's possible to connect this kind of line so you don't even approach the animal with the syringe or injection device, and it can be done automatically through the infusion pump at pre-selected time point.
Excellent. Out of curiosity, do you also have any experience with implantable pumps or like osmotic devices where they're preloaded with drugs and it's distributed through osmotic or process, or even a timed pump delivery as an implant? I'm assuming that would work just the same, but do you happen to have any experience with those?
Oh, yes, we do a lot of that kind of work, but mostly we are using osmotic pumps for more chronic, regular delivery of some compounds over time. Right. We haven't tried yet this kind of instantaneous release of large volumes or the concentrations of the compound during the imaging, and that would require some careful planning. So this is why the injections that are controlled by the operator through the external pump or the devices are much easier in terms of the process.
Excellent. That's a great clarification. Fantastic, gentlemen. Well, at this time, I'd say that brings us to the top of the hour and the end of our webinar. So thanks, Zsolt and Artem, for all your fantastic insights today, both in your presentations and certainly this Q&A session. And thank you, everyone in the audience, for joining and participating as well. So in closing, thank you again for taking part in this Inside Scientific webinar. And we look forward to having you with us again soon. Have a wonderful day, everybody.